Yes, they do. Gene duplication often lengthens the genome, adding more spaces for selection to occur. Celeste J. Brown, Christy M. Todd, and R. Frank Rosenweig and the Department of Microbiology, University of Idaho, performed experiments where after 450 generations of baker's yeast in a nutrient-limited environment, multiple tandem duplications occurred and developed into additional genes that allowed the yeast to make better use of the available environment for survival. Gene duplication is a mutation and an observed phenomenon. J. Zhang, Zhang Y.P., and H. F. Rosenberg published a paper in 2002 describing experiments where they observed the gene duplication of leaf-eating monkeys that resulted in the establishment of RNase 1b, that is an improvement over the RNase 1 enzyme in the monkey's digestive tracts. Overall, gene duplication is very common, with Haemophilus influenzae having 284 duplicate genes, to Homo sapiens having 15,343 duplicate genes. Duplicate genes, like in the example of the RNase 1b enzyme, can emerge from the selection pressures on the new duplicates, resulting in new information. First, it should be pointed out that monkeys are a broad category, like birds. This word encompasses many different species. As it turns out, our nearest relative is the chimpanzee, whose common ancestor we branched off from about 4 to 6 million years ago. Both chimpanzees and humans have a common ancestor, and we've since taken two different evolutionary paths. Evolution is an ever-branching tree, not a ladder of advancement. The question is like asking, if Americans came from Europe, why are there still Europeans? Not all of them migrated to the New World. That's what happens when a group splits. They geographically isolate from one another and diverge genetically, like what happened between chimps and humans. Irreducible complexity has been thoroughly refuted. It's the assertion that some structures, like the bacterial flagellum, will stop functioning even if one part is removed, and thus evolution, with successive additional steps, could not arrive at the functional flagellum. It must have been assembled with all parts at the same time. This couldn't have happened by blind chance, but rather deliberately done so by an intelligent designer. Irreducible complexity relies on an oversimplification of evolution. For instance, it ignores the possibility of a change of function. The structure may be irreducibly complex for one function, but not another. It also assumes that evolution cannot simplify or remove structure. With a combination of advancement of complexity, change of function, and reduction of complexity, even the bacterial flagellum can be arrived at through iterative steps. Stone arches are built in this way. A pile of dirt is built up, the stones are placed on top, producing a non-irreducibly complex system, then the underlying dirt is removed. Natural stone arches form in a similar way, where erosion burrows out the underlying rocks, leaving the irreducibly complex structure intact. All asserted examples of irreducibly complex forms in biology are refuted, and irreducible complexity is ultimately nothing more than an argument from ignorance. Even if it were true, it could simply be that some other natural mechanism is responsible for accounting for the phenomenon. Just because the current evolutionary theory doesn't account for a problem doesn't mean one is justified in merely making up an intelligent agency to explain it, void of any positively supporting evidence. Actually, all evidence indicates that radiometric dating is very reliable. Using several examples, we can show that the decay rates are consistent. First, for the duration that we've known about radioactive decay since the early 1900s, it's never changed. In the more immediate past, we can test on items, several hundred years to several thousand years old, that corroborate their approximate creation dates, and we independently know their historical timelines. Going further back in time, we can confirm that decay rates were the same 169,000 years ago. Supernovae produce radioactive isotopes that decay. Experimentation has found that different decay rates of these isotopes produce different wavelengths of gamma rays. Supernova SN197A is 169,000 light-years away, and thus, we can examine the wavelengths of the gamma ray lines. If the decay constants changed, the gamma ray lines would be off. They aren't, which means 169,000 years ago, the decay constants of these isotopes are unchanged. This same process finds a consistent pattern with supernova SN1991T, which is 60 million light-years away, meaning the constants were unchanged 60 million years ago. Going back even further, we find that the dating methods are consistent with the Okla natural fission reactor of about 1.8 billion years ago. What's more, many of these dating methods are independently verifiable with other dating methods, like ice core dating and dendrochronology. While radiometric dating isn't infallible, if the procedure wasn't done right, for example, typical objects are dated by multiple independent means, which minimizes potential error. One attempt at dating might have error, but the chances of multiple independent dating methods all failing in the same way is minuscule. So no, it's not an assumption, not when it's backed up by evidence. The only way to tell whether something is designed is through demonstration. The problem with this argument is an interfering bias of knowledge. Since childhood, we've been told that cars and houses are built by humans. At no point did we do an objective analysis of a car and ascertain that it was designed. We either observed it being designed, or we were told so. This problem can be brought into contrast if we change the question. Let's say that we have an unknown object X. How can we tell whether it's designed by examining its features? Does complexity, or size, or geometric patterns identify design? In order for us to determine the indicators of design, we'd have to compare designed versus non-designed objects. Then we should be able to find consistent attributes to distinguish the two groups. Here's the problem. Creationists and scientists don't agree on what's designed. I think plants and animals and humans aren't designed. They do. We can't begin to compare unless we settle on what belongs in each group. We also have to be careful that we aren't comparing organic versus non-organic instead of designed versus non-designed. We may accidentally be determining attributes of organic life instead of design. I don't know whether plants and animals are designed. I don't want to make that assumption. So the question is, how can we tell? Determining design through attribute analysis cannot work without making assumptions that beg the question. The only other possibility for perceiving design is some kind of extrasensory perception that creationists would have to demonstrate before it's a cogent possibility. Ultimately, we only have the first ability to know, through observation and education. In order to know that plants and animals are designed, creationists would have to demonstrate the creator creating them. That's what the argument from design is attempting to demonstrate in the first place. In the end, it's a circular argument.